All right, first thing I'm going to talk about is the nervous tissue. So this chapter is all about neurons and glial cells. And specifically for this section, I want to talk about neurons and kind of the organization of the nervous system with its structures and its functions. So first thing that you'll note in the notes is that we're talking about three functions of the nervous system. Particularly the tissue. So it's important for collecting info through receptors. So receptors are going to be anything that are receiving some sort of stimulus and then sending sensory signals to the spinal cord and brain to let them know, hey, this is happening. The second part is going to be the processing and evaluating of that information by the brain and spinal cord to determine how it's going to respond. So after that, effector cells are going to be in charge of actually responding to that specific stimulus. So the nervous tissue is responsible for initiating info through the brain and spinal cord, sending motor output via nerves to effectors. So we've got sensory signals that are coming in, the brain and spinal cord going ahead and thinking that through, and then we've got motor output via nerves to effector cells like muscles or glands actually trying to respond to that stimulus. So those are the three major functions of the nervous system. All of us should feel fairly comfortable with that. It's really more just recognizing that receptor cells are those sensory signals, and then the spinal cord and brain are going to be that central nervous system where things are really going to be decided on what's going to happen next, and then that we have effector cells that are going to be like muscles or glands that are going to go ahead and have some sort of motor output via nerves to go ahead and change something. For example, like if we're sweating or shivering in response to a change in our body temperature. Okay, so the nervous system it can be split into two different systems, the central nervous system and the peripheral nervous system. So a lot of times we'll hear CNS and PNS. So the central nervous system is going to include the brain and the spinal cord, and the peripheral nervous system is going to include the nerves and the ganglia. So nerves are just bundles of fibers that are actually just the axons coming from neurons. And then the ganglia are going to be clusters of cell bodies that are coming from these neurons that are making up the nerves. So a lot of times when we think of a nerve, we may just think of, oh, it's just like one neuron, but it's actually multiple stacked on top of each other, and that's going to be what's making up the peripheral nervous system. Of course, the peripheral nervous system is going to be the rest of our body. So we've got that central spinal cord and brain, and then everything coming off of it is going to be those nerves and ganglia. So some functional organization outside of just the nervous system being put into the central and the peripheral nervous systems, we've got this input and this output portion of the functional organization of the nervous system. So one of the inputs, or the input, is the sensory nervous system. Sensory nervous system can go ahead and be split into two different types. So we have the somatic sensory and the visceral sensory. So somatic is going to consciously perceive things from receptors. So those are going to be our eyes, our ears, and our skin. So we're going to notice those things consciously. The visceral sensory is going to be those things that we don't notice consciously. So it's going to be that sensory input that is unconsciously perceived, but you're still going to have receptors picking it up. So like blood vessels and internal organs, our heart, those different kinds of things that are happening all the time in the background that we don't necessarily notice is going to be the visceral sensory. On the output side, we have the motor nervous system. So the motor nervous system is responsible for it's the effector cells, right? So muscles and glands. So again, we have the somatic motor system, which is going to be consciously or voluntarily controlled. So that's going to be like skeletal muscles. So me moving right now and talking right now is the somatic motor nervous system where I'm consciously choosing to do those things. On the other side, we have the autonomic motor system. I just kind of think of like if you're wanting autonomy, right? You want to be separate from your parents and be able to make your own decisions. So in that regard, autonomic motor system is unconsciously controlled. So we don't necessarily keep tabs on that all the time, so it's involuntary. Effectors are like cardiac muscle, smooth muscle, and glands. So when your hypothalamus goes ahead and produces different glands, or when your smooth muscle goes ahead and contracts to cause peristalsis to bring food down, we're not consciously making those decisions, so it's going to be our autonomic motor system. All right, on to the next page of notes. This is all focused on neurons. So neurons are pretty awesome. They have 5 different specific kind of characteristics that you can remember. Neurons are going to be excitable, so they respond to a stimuli, so chemical, stretch, pressure change. There's even some, the cool thing is that just like pushing on your own skin, what's actually causing you to feel those things is because we have neurons that are picking that up just by the pressure change. So stimulus changes the resting membrane potential, which means that there's actually a gradient, a concentration gradient that's causing a voltage in between the inside of the neuron and the outside, so in the interstitial fluid around the neuron. And so that's an important part for the excitability, and we'll learn more about that. The next part is conductivity. So that means an electrical change can quickly go or propagate along the plasma membrane inside of the neuron as voltage-gated channels are open during an action potential. So basically, we're just kind of seeing this movement of charge all the way down the axon or through the dendrites or through the soma or the cell body of the neuron. And it just is able to conduct that all the way down, just like a piece of copper wire could in your housing for electrical. The third part is going to be secretion. So neurons are capable of releasing neurotransmitters in response to a conductive activity. So that conductivity that we just talked about is going to move along the cell and cause these vesicles to go ahead and be released. And they can excite the cell to continue to propagate that conductivity, or it can inhibit it, so it'll stop some. So it's going to have an effect on those neurotransmitters are going to have an effect on other neurons or on effector cells like glands or muscles. The fourth one is that they can live a really long time. Neurons, most of them have been formed during our fetal development in the womb, and they're actually still functional until we're very, very old. So it's pretty amazing that those cells don't divide, they just live forever. So within that, the last part that makes them special is that they're amitotic. So remember in Latin that a just means without or doesn't have. So it doesn't go through mitosis. So during fetal development, neurons, their mitotic activity is lost in most of them, except those in the olfactory epithelium of the nose and in certain areas of the brain. So some pieces of the brain you're going to find mitosis happening in the neurons and in our nose as well. But other than that, they pretty much just are made, and then we don't get any new ones. Okay, so one of the things here, I went ahead and drew a picture for you guys of the neuron connecting with another neuron, and so this picture is uploaded onto Summit, so hopefully you're following along with that. So the neurons, one of the biggest pieces is our dendrites, our soma or the cell body, and then the axon. And as we kind of think about those, really the most important part is that the dendrites are going to be receiving an electrical impulse input from a different neuron or a effector cell sensing or something along those lines. And so they're receiving information and they're non-myelinated. So that means that they don't have any sort of glial cell wrapped around them to make sure that the conduction of electricity happens quickly. They're just receiving information. In the axon, you've got this little portion called the axon hillock that's going to go ahead and help propagate a membrane potential down the axon if it's large enough, and you'll learn more about that coming up. So the axon is important for conducting action potentials and releasing neurotransmitters at the very end of the axon. So at the end of the axon, we have these things called synaptic knobs, and so you'll see them right here. Actually, they're connecting to another neuron, and that's where you're going to find just this cleft there that's a space for the neurotransmitters or electrical charge to go ahead and come across to go into the next neuron to continue to travel down. So you want to spend some time on your own just looking at the different pieces. This is much more about the anatomy of a neuron. So you'll want to make sure that you understand those different pieces there and have those memorized. So neurons have a really high tensile strength. That means that they can you can pull on them and they're going to stay together just like a steel cord or cable. And the reason for that is their cytoskeleton is made up of these neurofilaments, or just bundling of neurofibrils that are going to be microtubules and different proteins that are very strong. So one of the things that we also note is that, okay, dendrites are going to receiving input, and then the axon, the end of the axon, or the telodendria at the very end, they're going to go ahead and continue that. They're going to do the output portion of the neuron's conductivity. And so there's two different terms we use for that. There's anterograde transport, which is going to be things traveling from the cell body down. So that's going to be like an action potential or information moving down, so new materials. And then so coming down, and then we also have this other stuff that's going to be coming up towards the cell body, and we call that retrograde transport. So old materials for recycling, right? So if we've got something that's just old and broken, it's going to go back up to the soma or the cell body and be worked on there, because that's where you're going to find all those different organelles like the lysosome and the nucleus and the Golgi apparatus. So both of these anterograde and retrograde transports are going to happen either by fast axonal transport or slow axonal transport. So axonal just means up or down the axon, right? Fast axonal transport is 400 millimeters per day, and it has to have ATP, and it just moves along these microtubules that are making up the cytoskeleton. And it can either be antero, so coming down from the soma, or retro, where it's going back up towards the soma. It's just way faster, and it requires ATP, so it needs energy. The slow axonal transport is going to be by the axoplasm. So just like cytoplasm, the axon is also going to have this fluid within it, and it moves way slower. It's going to be 0.1 to 3 millimeters per day. I mean, it doesn't use ATP, so it's free flow, which is why it's so slow. It's just kind of going with this axoplasm that's within the axon. And it only goes anterograde, so it's only going to move down. It's not going to go against the flow of the nerve impulse as it travels down the dendrites into the soma and down the axon. All right, a couple last pieces here. So we've got two more portions. This is probably one of the biggest portions of this chapter, just talking about neurons and their different shapes, and then talking about nerves. So we can structurally classify neurons as either multipolar, bipolar, unipolar, or anaxonic. So multipolar just means that you're going to find these are the ones that we see most of the time, is that we're going to have these neurons that have dendrites at the end of the cell body, and then it's going to come down into that axon. Actually all motor neurons are going to be made up of this multipolar, along with most interneurons. So interneurons we find within our central nervous system. So this is the one that we're most used to like drawing or seeing. Bipolar are interesting. They've got a cell body in the center, or a soma in the center, and then we've got the dendrites here, and then the axon on this side. So this is actually not super common. It's more special sense nerves that are going to be having that, and you're going to find a collection of these cell bodies called the ganglia right next to the spinal cord. The next one we have is the unipolar. So you'll see that it's got the dendrites and the sensory the dendrites here, and then dendrites here, and we've got this axon here in the middle with the peripheral and a central portion, and this is most sensory neurons, so they kind of have that similar structure to this guy over here, but they've got these different portions, and the axon is actually right there in the middle, so we call those unipolar. The anaxonic just means that they have no axon. They're just going to be receiving information through the dendrites. And these ones are going to be these interneurons that we find in the central nervous system. Not as many as these, multipolar ones, but they're going to be found also within the brain. Okay. So that's some of the structural differences. When we talk about the functional classification, we've got three different types. So we have sensory neurons, motor neurons, and interneurons. Sensory neurons are afferent. They're going to pick up information, okay? So they're affected by something else, and they're going to transmit that to the central nervous system. Typically are going to be unipolar or bipolar. So those were the two sensory ones that we looked at in the middle, with the cell body or the soma in the middle of the axon and dendrites. Then we have our motor neurons. These are efferent neurons, and they take info from the central nervous system to somatic or visceral effectors and are going to cause something to happen. So they're multipolar. Those are the ones that we're used to seeing. The last one is the interneurons. They actually make 99% of our neurons, and we call them association neurons. So they're going to be in between a sensory neuron and a motor neuron, just hanging out there in the middle, and they're going to receive, process, and integrate info from all these other neurons within the spinal cord so that they can communicate. So those motor and sensory nerves can communicate between them. And then in the central nervous system, they're going to be multipolar or anaxonic, so in the spinal cord or in the brain. So neurons and nerves, kind of already mentioned this, but nerves are just a bundle of parallel axons in the peripheral nervous system. They have connective tissue wrapping all the way around it, just kind of like the muscle does. So you're going to have the endoneurium, which is going to cover one axon. Then you're going to have the perineurium that's going to cover a small bundle of axons, just like the fascicle that we find in muscle tissue. And then the epineurium is going to cover the entire nerve. So we're going to have a couple different bundles of these nerve cells that are wrapped around with perineurium, and then all of it's going to be wrapped around with epineurium. So there's a couple types of nerves. The structural classification is going to be cranial versus spinal nerves. And then our functional classification is going to be sensory nerves, motor nerves, and then mixed nerves. So you might have nerves that both in that bundle wrapped around with epineurium may have both sensory and motor nerves. So synapses between these neurons are going to connect each other. So there's just a little synapse between one neuron and another that can be either electrical or chemical. So these nerves are going to have lots of these neurons collected in there, and they're either going to send an electrical signal or a chemical signal. So the electrical are super fast. There's no delay. The neurons are connected by gap junctions, and it just goes right across. Chemical is a little slower, but it's also more common. So neurotransmitters are going to be the most common thing that you're going to find in those little vesicles at the, what do we call those? The telodendria in the synaptic knobs at the very end of the axon. So the presynaptic axon terminal creates the signal, so up at the top or at the end of the axon, and then it's going to go ahead and connect with the postsynaptic area, which is going to be on the soma typically, or on the dendrites of that cell. Usually it's going to be the dendrites. So it's going to receive those signals. There's a cleft in between there, the synaptic cleft, and it's just small and fluid filled between the two neurons. The neurotransmitter is going to be released in vesicles, so little bubbles, and they're going to go move through that synaptic cleft into the postsynaptic area. It's going to diffuse across there, bind to the postsynaptic receptors, and when it binds, it's going to cause a potential. So it's going to cause channels to open, and there's going to be change in a concentration gradient. That whole process there is called the synaptic delay, and it's just the time that it takes for those events to occur. So it's not immediate like electrical. It has a couple steps to go through for the chemical communication. And that is it for this section.